Good morning, children. It is in your hands to create a better world for all who live in it. Okay. Before going to the lesson breeding and biotechnology, let us see recap what we have learned so far. Methods of plant breeding for crop improvement. In that we studied the following headings such as introduction of new varieties of plants, selection, polyploidy breeding, mutation breeding, and hybridization. So these are all the things that we have learnt. Today we are going to study, these are the things we are going to study. They are animal breeding, inbreeding, outbreeding, heterosis, genetic engineering, techniques of genetic engineering, gene cloning, biotechnology in medicines, stem cells, DNA fingerprinting technology, genetically modified organisms. Now let us start with animal breeding. First of all, we should know animal husbandry. What do you mean by animal husbandry? Animal husbandry involves the breeding of animals. It aims at improving the genotypes of animals to make them more useful to the welfare of mankind. This emphasizes domestication and propagation of animals under controlled conditions to enhance food production and food quality. Then, what do you mean by a breed? A breed is a group of animals of common origin with a species, within a species that has certain distinguishing characters that are not found in other members of the same species, like general appearance and other striking features. Then, breeding. Breeding involves mating patterns of different varieties, each having some desired trait which are passed on to the offspring. Okay, next we pass on to objectives of animal breeding. Animal breeding aims at improving the genotypes of domesticated animals to increase their yield and improve the desirable qualities to produce more milk, egg, and meat. When breeding takes place between animals of the same breed, it is called inbreeding. For example, inbreeding means cross made between the same breed among the sheep variety. And outbreeding means a cross made between the donkey and the horse, that is different breeds. This is called outbreeding. And now let us pass on to inbreeding. Inbreeding refers to the mating of closely related animals within the same breed for about 4 to 6 generations. Superior males and superior females of the same breed are identified and mated in pairs. It helps in the accumulation of superior genes and the elimination of genes which are undesirable. One example is given, Hisardale. Hisardale is a new breed of sheep variety developed in Punjab by crossing Bicanary Eves and Australian Merino rams. So here Hisardale is a breed among sheep variety. So this is called inbreeding. So inbreeding depression. What do you mean by inbreeding depression? Continued inbreeding reduces fertility and productivity. Then if we make a cross among same variety, finally it reduces the fertility, and fertility means cannot reproduces. So it reduces fertility and productivity. Inbreeding exposes harmful recessive genes that are eliminated by selection. Children, I told you Bicanary Eves are Indian breed of wool type sheep having the body white and the face usually colored. And the Merino is one of the most historically relevant and economically influential breed of sheep, much praised for its wool, that is Australian merino rams. So if we make the cross between these two, we can get a Hisardale. A Hisardale is a new breed of sheep developed by Indian and Australian cross. Next we pass on to outbreeding. It is the breeding of unrelated animals. The offsprings formed are called hybrids. The hybrids are stronger and vigorous than their parents. A cross between two different species with desirable features of economic value are mated. Then if we make a cross between a male donkey and a female horse, the hybrid is called mule. Mule is superior to horse in strength, intelligence, ability to work, and resistance to diseases, but they are sterile. What is the meaning of the word sterile? Sterile means we cannot reproduce. It lacks the capacity of reproduction. So next we pass on to heterosis. Heterosis is the superiority of the hybrid obtained by crossbreeding is called heterosis or hybrid vigor. Heterosis means if we mate two unrelated species, unrelated animals, the hybrid has the superior character over the parents is called heterosis or hybrid vigor. Then we pass on to effects of hybrid vigor in animal breeding. Increased production of milk by cattle. Then increased production of egg by poultry. High quality of meat is produced. Increased growth rate in domesticated animals. See, we are increasing the production of milk, egg, and the meat by the production of more production of animals, because to meet the increased population. Children, next we pass on to genetic engineering. Genetic engineering is the manipulation and transfer of genes from one organism to another organism to create a new DNA called recombinant DNA or rDNA. The term recombinant is used because DNA from two different sources can be joined together. Hence, genetic engineering is also called as recombinant DNA technology. Children, now in genetic engineering, we come across different terms such as plasmid, restriction enzymes, and genetic engineering tools we come across. Let me explain. Plasmid is the small circular double-stranded DNA molecule found in the cytoplasm of a bacterial cell and separated from chromosomal DNA. It can replicate independently. It is called plasmid. Then what do you mean by restriction enzyme and DNA ligase? Restriction enzymes used to cut the DNA segments at specific sites, so they are called molecular scissor. So in genetic engineering, we come across these enzymes. Enzymes are nothing but biocatalysts which speed up the chemical reaction. So this restriction enzyme and DNA ligase are used as genetic engineering tools. Okay, now let us pass on to techniques of genetic engineering or basic requirements. Important discoveries that led to the stepping stone of rDNA or recombinant DNA technology were the presence of plasmids. What do you mean by plasmid? Just now only I explained you. Plasmid is the small circular double-stranded DNA molecule. So presence of plasmid in bacteria that can undergo replication independently along with chromosomal DNA. There is a difference between the chromosomal DNA and the circular double-stranded DNA. Then restriction enzyme. Restriction enzyme is otherwise called molecular scissors. So restriction enzyme cuts or break DNA at specific sites and are called as molecular scissors. Then DNA ligase. DNA ligase are the enzymes which help in joining the broken DNA segments. So DNA ligases are called pasting enzyme. Restriction enzymes otherwise called molecular scissor. DNA ligases are called pasting enzyme. Children, now we pass on to gene cloning. What do you mean by gene cloning? Gene cloning is the process in which a gene of interest is located and copied of DNA extracted from an organism. When DNA is extracted from an organism, all of its genes are extracted at one time. This DNA, which contains thousands of different genes. So the carbon copy of an individual is often referred as clone. However, more appropriately, a clone means to make a genetically exact copy of an organism. So in gene cloning, a gene or a piece of DNA fragment is inserted into a bacterial cell where DNA will be multiplied, or we can make copies of DNA as the cell divides. Now let us pass on to basic steps involved in gene cloning. Isolation of desired DNA fragment by using restriction enzyme. Restriction endonuclease is also called a molecular scissor that cut DNA fragment at any specific site. Then insertion of DNA fragment into a suitable vector. Vector is already I told you plasmid to make our DNA, recombinant DNA. So we have to insert this specific fragment of DNA into the plasmid suitable vector. Then transfer of this rDNA into the bacterial host cell. Bacterial host cell means if we want to introduce this DNA segment into the bacteria. Why we select bacteria? Because bacteria only multiply very fast. So only we select bacteria. So transfer of rDNA into the bacterial host cell. That process of this transfer of rDNA into the bacterial cell is called transformation. Then selection and multiplication of recombinant host cell to get a clone. Clone already I told you. The carbon copy of an individual is called clone. Then expression of a cloned gene into the host cell. Then we have to introduce into the host cell. So using this strategy, we can produce several enzymes, hormones, and vaccines. You know what do you mean by enzyme? Enzymes are biocatalysts which speed up the chemical reactions. Then hormones. Hormones are secreted by the endocrine glands. Sometimes hormones also, if hormones are not secreting in a particular person, we can introduce this type of gene cloning we can do. And vaccines. Vaccines are nothing but to treat any diseases or before the precautionary measures we use vaccines. So using this strategy we can produce enzymes, hormones and vaccines. Children, now we pass on to development of Dolly. Dolly was the first clone produced from a cell taken from an adult mammal. The production of Dolly showed that genes in the nucleus of such a matured differentiated somatic cell are still capable of producing an embryonic totipotent state, creating a cell that can go on to develop into any part of an animal. What do you mean by totipotency? Totipotency means the ability of a single cell to divide and produce all of the differentiated cells in an organism. Dolly is the name of a sheep that has the honor of being the first mammal to be cloned by a group of scientists at Roslin Institute, Scotland. Dolly was born on 5th July 1996 and she passed away in 2003 because of lung disease. She lived for 6.5 years as a normal active sheep. Children, now we pass on to biotechnology in medicine. Using genetic engineering techniques, medicinally important valuable proteins are polypeptides that form the potential pharmaceutical products for the treatment of various diseases have been developed on a commercial scale. Children, what do you mean by polypeptide? So many proteins are together form polypeptide chain. And pharmaceutical products developed by recombinant DNA technique. Insulin, you all know, people, those who are suffering by diabetes, they use insulin. So insulin also we can produce by biotechnology method. So insulin used in the treatment of diabetes. Human growth hormone used for the treating children with the growth deficiency. Some children, they have the growth deficiency because of the less production of hormones. So in such cases, we can introduce this growth hormone into the children. And blood clotting factors are developed to treat hemophilia. What is the disease called hemophilia? Hemophilia is usually an inherited bleeding disorder in which blood does not clot properly. Then tissue plasminogen activator is used to dissolve blood clots and prevent heart attack. See, tissue plasminogen activator is a protein involved in the breakdown of blood clots. Then development of vaccine against various diseases like hepatitis B and rabies. Children, now we pass on to gene therapy. What do you mean by gene therapy? Gene therapy is an experimental technique that uses genes to treat or prevent disorders. In the future, this technique may allow the doctors to treat disorder by inserting a gene into a patient cell instead of using drugs or surgery. So the gene therapy refers to the replacement of defective genes by the direct transfer of functional genes into humans to treat genetic disease or disorder. The genetic makeup of the patient cell is altered using recombinant DNA technology. It was first successfully implemented in 1990. There are two types of gene therapy: somatic gene therapy and germline gene therapy. Somatic gene therapy is the replacement of defective gene in the somatic cell. Germline gene therapy is the replacement of defective gene in the germ cell, egg and sperm. Gene therapy conducted till date targeted only somatic cells. Correction of genetic defects in somatic cells may be beneficial to the patient, but the corrected gene may not be carried in the next generation. Now let us pass on to stem cells. What is actually stem cells? Stem cells are special human cells that are able to develop into many different cell types. This can range from muscle cells to brain cells. Our body is composed of over 200 specialized cell types that can carry out specific functions. Example, neurons or nerve cells that can transmit signals, or heart cells which contract to pump blood, or pancreatic cell to secrete insulin. These specialized cells are called differentiated cells. In contrast to differentiated cells, some cells are undifferentiated or unspecialized mass of cells. The stem cells are the cells of variable potency. Potency refers to the number of possible fates that a cell can acquire. The two important properties of stem cells that differentiate from other cells are ability to divide and give rise to more stem cells by self-renewal. Second one, its ability to give rise to specialized cells with specific functions by the process of differentiation. Why the stem cells are important? Stem cells represent an exciting area in medicine because of their potential to regenerate and repair damaged tissue. Some current therapies such as bone marrow transplantation already make use of stem cells and their potential for regeneration of damaged tissue. Okay, now let us pass on to types of stem cells. There are two types of stem cells such as embryonic stem cells and adult stem cell. Embryonic stem cells can be extracted and cultured from the early embryos. These cells are derived from the inner cell mass of the blastocyst. What do you mean by blastocyst? It is nothing but is a structure formed in the early development of mammals. It possesses an inner cell mass which subsequently forms the embryo. These cells can be developed into any cell in the body. Then adult stem cell or somatic stem cell. Adult stem cell or somatic stem cells are found in the neonatal and adults. Neonatal means a newborn baby. They have the ability to divide and give rise to specific cell types. Sources of adult stem cells are amniotic fluid, umbilical cord, and bone marrow. Children, what do you mean by bone marrow? Bone marrow is the spongy tissue inside some of your bones such as hip and thigh bones. It contains stem cells. The stem cells can develop into red blood cells that carry oxygen through your body. The white blood cell that fight infections and platelets that help with blood clotting. Now let us pass on to stem cell therapy. Sometimes cells, tissues and organs in the body may be permanently damaged or lost due to genetic condition or disease or injury. In such a situation, stem cells are used for the treatment of diseases which is called stem cell therapy. In treating neurodegenerative disorders like paralysis, Parkinson's disease and Alzheimer's disease, neuronal stem cells can be used to replace the damaged or lost neurons. Children, what do you mean by Parkinson's disease? It is a progressive disease of the nervous system marked by the tremor, muscular rigidity, and slow then impressive movement chiefly affecting middle-aged and elderly people. It is associated with the degeneration of basal ganglia of the brain. Then what do you mean by Alzheimer's disease? A progressive disease that destroys memory and other important mental functions. Brain cell connections and the cells themselves degenerate and die, eventually destroying memory and other important mental functions. Chalo, now we pass on to DNA fingerprinting technology. What is actually DNA fingerprinting technology? DNA fingerprinting is a laboratory technique used to establish a link between biological evidence and a suspect in a criminal investigation. And this DNA profiling is the process of determining an individual's DNA characteristics. DNA analysis intended to identify a species rather than an individual is called DNA barcoding. The human genome has 3 billion base pairs. Did you know that the DNA pattern of two individuals cannot be same except for identical twins? Each person's DNA sequence is unique due to small difference in the base pairs. Therefore, if you want to compare the genetic difference among the two individuals, DNA fingerprinting is easier and quicker method. This technique was developed by Alec Jeffrey. The technique analyzes each individual's unique DNA sequences and provides distinctive characteristics of individual which helps in identification. Then variable number of tandem repeat sequence serve as molecular markers for identification. It is shortly called as VNTRS. In human beings, 99% of the DNA base sequence are the same, and this is called as bulk genomic DNA. The remaining 1% of DNA sequence differs from one individual to another. This 1% of DNA sequence is present as small stretch of repeated sequence which is known as satellite DNA. The number of copies of the repeat sequence, also called variable number of tandem repeat sequence, differs from one individual to another and results in variation in the size of the DNA segment. The satellite DNA consists of very large layers of tandemly repeating non-coding DNA. Satellite DNA is the main component of functional centromeres and form the main structural constituents of heterochromatin. As shown in the illustration, the sequence of AGCT is repeated 6 times in the first person, 5 times in the second person, and 7 times in the third person. Because of this, DNA segment of third person will be larger in size, followed by DNA segment in the first person and then the second person. Thus, it is clear that satellite DNA, that already I told you, the satellite DNA bring about variation within the population. Variation in DNA binding pattern reveals differences among the individual. Children, now we come across a term called variable number tandem repeat. This variable number tandem repeat is a location in a genome where a short nucleotide sequence is organized as a tandem repeat. This can be found on many chromosomes. Chromosomes often show variation in length, that is a number of repeats among the individuals. Children, this variable number of tandem repeats are short nucleotide sequence with a number of 20 to 100 base pairs that vary in copy number in bacterial genomes. They are thought to be arise through the DNA strand during replication and are of unknown function. Then applications of DNA fingerprinting. DNA fingerprinting technique is widely used in forensic applications like crime investigations such as identifying the culprit. It is also used for paternity testing in the case of disputes. It also helps in the study of genetic diversity of population, evolution and speciation. Children, just now I told you DNA paternity testing. What is this DNA paternity testing? It is the use of DNA profiles to determine whether an individual is the biological parent of another individual. Paternity testing can be easily important when the rights and duties of the father are in issue and the child's paternity is in doubt. Children, now we pass on to genetically modified organisms, shortly called GMOs. One of the most tremendous development of genetic engineering is the production of genetically modified plants and animals. The genetic modification refers to the alteration or manipulation of genes in the organism using recombinant DNA technique in order to produce desired characteristics. The DNA fragment inserted is called transgene. See, transgenic animals are animals that have had the foreign gene deliberately inserted into their genome. Such animals are most commonly created by the micro-injection of DNA into the pronuclei of the fertilized egg, which is subsequently implanted into the oviduct of a pseudo-pregnant surrogate mother. So these plants are animals expressing a modified endogenous gene or foreign gene are known as transgenic organism. The transgenic plants are much stable with improved nutritional quality, resistant to diseases, and tolerant to various environment conditions. Similarly, transgenic animals are used to produce proteins of medicinal importance at low cost and improve livestock quality. Some examples are genetically modified plants and animals are given in the table below. Now let us see some genetically modified plants. Its objective is improved nutritional quality in rice. The gene inserted is beta-carotene gene. Usually in humans, beta-carotene is required for the synthesis of vitamin A. Then what is the achievement? Golden rice. We discussed in the previous classes. Golden rice, genetically modified rice, can produce beta carotene that can prevent vitamin A deficiency. Then increased crop production. Bt gene is nothing but from bacteria Bacillus thuringiensis. It is called Bt. Bt gene produces protein that is toxic for insects. Insect resistant plants. These plants can produce the toxin protein that kills the insects which attack them. Now, genetically modified animals. In animals such as in fishes and in the sheep, we can introduce a genetically transgene. So, the improved wool quality and production. What is the gene inserted? The gene for synthesis of amino acid cysteine. Cysteine is one of the amino acid. Then the sheep developed is called transgenic sheep. And in the same way, this genes can introduced in the fishes such as rainbow trout and tilapia. Growth hormone gene can be introduced in such fishes for increased growth in fishes. Then these fishes are called transgenic fishes.